Welcome back to The Explainer, everyone. I am incredibly excited to jump in today because we're tackling one of the most fundamental, elegant, and frankly, fascinating engineering problems in all of cell biology. We're going to look at why cells are the size they are and why they can't just, keep growing indefinitely. If you've ever wondered why we're made of trillions of microscopic cells instead of just a few massive ones, well, you are in the exact right place. We are going to break down the specific mathematical paradox that dictates the literal limits of life. Okay, let's dive right into a little thought experiment. Imagine a single cell the size of an actual basketball. Now, at first glance, a giant amoeba like this might seem like an unstoppable powerhouse, right? But look at those tiny blue vector arrows, representing microscopic oxygen and nutrients, trying to get inside. Notice how they completely fail to reach the core? In a basketball-sized cell, these essential molecules have to travel inches instead of micrometers. By the time they diffuse even a fraction of the way in, the center of the cell is completely starved. It's an absolute biological disaster. And to understand exactly why this happens, we've got to look at the geometry. So here is our roadmap for dismantling this mystery today. First, the basketball cell problem. Second, unlocking the core math. Third, calculating the exact ratio drop. Fourth, biological shape adaptations. And finally, exam tips and a recap. All right, let's start with section 1, the basketball cell problem, and define the exact engineering bottleneck we're dealing with here. Let's visually isolate the outer boundary of our cell from its inner core. The concept ruling this entire domain is the surface area to volume ratio, or SAV ratio. This is basically a critical measure of how efficiently a cell can exchange stuff with its environment. Look at those directional vectors. The green arrows pulling inward show the intake of oxygen and nutrients through the surface area. At the same time, you've got red arrows pushing outward, expelling waste. Think of the surface area as the cell's only doorway and the volume as this massive, bustling factory floor inside that requires constant supplies and generates constant waste. If that factory floor gets too big but the doorway stays relatively small, the whole system just backs up and fails. So why exactly does that doorway become too small as the cell gets bigger? Let's move to section 2, the core math explained. Now, what's really wild is how we can actually see this geometric trap unfold. Let's watch the mechanics step by step. Step 1, to find the surface area of a sphere, the formula is 4 pi r squared. Notice how the radius, r, is squared? That is two-dimensional growth for the boundary membrane. But now look at step 2, the volume. The volume of a sphere is 4/3 pi r cubed. Cubed! That's a three-dimensional growth for the inside of the cell. Because a cube grows astronomically faster than a square, the volume just explodes in size while the surface area totally lags behind. So in step 3, when you divide the surface area by the volume to get our ratio, the math simplifies elegantly down to just 3/r. So here is the crucial bottleneck. As that radius increases, look at that dimension line stretching from the center. The overall ratio drops. As we drag that radius out, watch those inward-pointing nutrient vectors dynamically thin out, fading from bright green to a dull gray. It vividly shows how diffusion becomes agonizingly less efficient the larger the cell gets. The cell simply cannot feed its massive internal volume through its proportionally shrinking skin. But hey, we don't just want to talk about the theory. We want to prove it. Let's jump into section 3 and calculate this ratio drop side by side. Let's walk through this worked example. On the top row, suppose we have a cell with a truly microscopic radius of just 1 unit. Using our formulas, the surface area is 4 pi times 1 squared, which is 4 pi. The volume is 4/3 pi times 1 cubed, which gives us 4/3 pi. When we calculate our SAV ratio using that simplified 3/r formula, 3/1 gives us a nice healthy ratio of precisely 3. Now, let's look at the bottom row. We merely double the radius to 2 units. Doesn't seem like much, right? But the surface area becomes 16 pi, and the volume balloons to 32/3 pi. Our SAV ratio is now 3/2, which equals 1.5. Literally, by just doubling the size of the cell, the efficiency ratio has been brutally slashed exactly in half. And this chart brilliantly illustrates that undeniable plummet in efficiency. Bar 1 stands tall at a value of 3, but simply doubling the width violently cuts efficiency down to 1.5 in bar 2. That stark downward vector connecting the tops of the bars, that is the exact mathematical reason why a massive basketball-sized cell would starve to death. the diffusion mechanics simply break down. So, if the math is this immovable physical bottleneck, how does complex life exist at all? This brings us to section 4, biological shape adaptations, and how evolution essentially engineers its way around the math. First, let's clear up a massive misconception. A really common mistake is thinking that scaling up a cell's size increases its efficiency. But as we just mathematically proved, scaling up ruins efficiency. Instead, look at the biological reality here. Biology adapts the cell's shape to maximize surface area without massively increasing inner volume. We basically have to abandon the sphere entirely to solve this diffusion problem. Let's see how this actually builds into real-world biology. Notice the structural shifts from macroanatomy on the left to the microcellular solutions on the right. Plant root hair cells adapt by elongating. They push these long, thin protrusions right into the soil. This creates a massive surface area for absorbing water and minerals with barely any increase in internal volume. Now look at the lining of the small intestine. The membrane isn't flat. It is heavily folded into these microscopic peaks and valleys called villi and microvilli. These folds pack a staggering amount of surface area. Remember, those are our doorways, into a tiny space, allowing for incredibly rapid nutrient absorption. Evolution literally bends the geometry to beat the math. This is absolutely foundational stuff, guys. So let's move into our final section, exam tips and recap, so we can make sure you capture every single valuable mark on your upcoming tests. Let's do a quick-fire recap. Treat these bullet points as your ultimate study checklist. First, always remember the cardinal rule. The surface area to volume ratio drops as cells grow. Second, this decreased ratio directly limits diffusion efficiency. That is the explicit physical reason large single cells can't survive. Third, if an exam asks how cells overcome this, point directly to shape alteration. They elongate or they fold their membranes to maintain a high ratio. And finally, practice calculating that 3/r formula. Visualizing how changes in the radius impact that final efficiency number is absolutely going to secure you crucial marks on test day. We've seen how elongating and folding can basically cheat the physical limits of the sphere, allowing for the brilliant complexity of the human intestine or the sprawling network of plant roots. But it leaves us with a pretty incredible thought to ponder. If the math limits a simple sphere, what other crazy, complex geometric shapes could evolution unlock to push the boundaries of life even further? Could there be structural forms waiting to be discovered or entirely different ways of organizing volume that we haven't even dreamed of yet? Keep observing, keep questioning, and definitely keep studying those dimensions. Thank you so much for joining me on The Explainer today, everyone. I'll see you next time.